Hi everyone, my name is Taylor Landry and I'm a first year PhD student in Dr. Courtwright's bioenergetics class. He has challenged us to, via a PowerPoint presentation or video or any other method, to present the different compartments of the mitochondria and describe the specific processes and where they occur in each respective compartment. And so, for the sake of brevity, I will not go into detail of all these processes, but hopefully by the end of the video you'll understand at least the location of these processes, and you'll be able to picture that and imagine that as you go throughout your studies and learn more in depth of what's going on. And so, as you can see, I'm definitely not an artist, but on this board I've drawn a mitochondrion. So it's represented by this big oval surrounded by the outer mitochondrial membrane, and inside that big oval is a more squiggly shape, and that is the inner mitochondrial membrane. And we'll call everything outside of this oval, or outside of the mitochondria, the cytosol. And so it looks like there's a lot going on, but hopefully I'll be able to zoom in and kind of look at the different processes as a little snapshot and make it a little more simple. And so we'll start off kind of easy. We'll start off with the creatine phosphate reaction. And so creatine phosphate and ADP via creatine kinase can become creatine ATP, an important process for high intensity exercise and rapid ATP synthesis. And notice that this enzyme, creatine kinase, exists in the cytosol. However, as described by the creatine phosphate shuttle, creatine kinase can also exist in the inner membrane space, and it is essential for resynthesis of creatine phosphate so that this process can continue occurring. Another easy one, we have the myokinase or adenylate kinase reaction. It's a reversible reaction, and it involves the synthesis of two ADPs from ATP and AMP, or the synthesis of ATP and AMP from two ADPs. Notice once again, this ADK, the enzyme catalyzing the reaction, is in the cytosol. And so, if you've read the theoretical paper suggested by Dr. Courtwright, it's suggested that ADK actually exists in many other locations other than the cytosol, including the inner membrane space and the matrix of the mitochondria. The theory behind this is that AMP and myokinase, or ADK, are very sensitive energy signaling molecules and help regulate many bioenergetic pathways. And by being located in the different compartments of the mitochondria, it allows for communication between and within these compartments. So now that we've talked about the creatine kinase and the adenylate kinase reactions, let's get a little more complicated. And so let's talk about glucose metabolism. So glucose can come from glycogenolysis or gluconeogenesis or just in the diet, all of which occurs in the cytosol. Glucose to pyruvate, or in other words, glycolysis, also occurs in the cytosol. And here's where it gets a little more complicated. Pyruvate has many fates. Pyruvate can become lactate via lactate dehydrogenase in the cytosol. And both pyruvate and lactate can enter the mitochondrion via MCTs. Now, more research definitely needs to be done about LDH and lactate metabolism, and I'm sure you'll talk about it in depth in the bioenergetics course. But as far as we understand, there's actually lactate dehydrogenase in the inner membrane space, as well as in the matrix of the mitochondria. But I'll get to that. And so, once pyruvate enters the mitochondria, it can enter the matrix via many different mechanisms. So I talked about the MCT, but also you have the pyruvate and hydrogen ion symporter and the pyruvate and hydroxide ion antiporter. I also want to talk about a mechanism addressed by the Kane paper, and the Kane paper talks about this LDH in the inner membrane space, allowing for pyruvate to be converted into lactate, and then lactate as itself would enter the matrix of the mitochondria to become pyruvate, a phenomena called the intracellular lactate shuttle. I'm sure you've heard about the intercellular lactate shuttle, where this lactate would be transported to different tissues, whether it be the heart or other muscle, to be used as fuel. And so, now that our pyruvate and lactate are inside the mitochondria, the matrix of the mitochondria, note that pyruvate dehydrogenase, the conversion of pyruvate to acetyl-CoA, as well as the TCA cycle, occur in the matrix of the mitochondria. And so, there's many different fates of pyruvate as well as acetyl-CoA, and so let's just kind of work our way through the TCA cycle. Acetyl-CoA can become citrate via citrate synthase, and this citrate, an intermediate in the TCA cycle, can continue going through the TCA cycle, but also it plays an important role when it exits the mitochondria. It plays an important role in fatty acid synthesis. And so citrate via ATP citrate lyase can produce oxaloacetate and acetyl-CoA. And then acetyl-CoA via acetyl-CoA carboxylase can produce millennial-CoA and go through fatty acid synthesis. Also, I want to mention this acetyl-CoA can also be essential to ketogenesis in times of fast. But remember, it takes 2 or 3 days for the enzymes necessary for ketogenesis to undergo transcription and translation. So back to our TCA cycle. So here we have citrate, and it can go to this little 2 I have drawn. This 2 I'll address later, but it represents succinate dehydrogenase as well as complex 2 of the electron transport chain. This 2, unlike the rest of the TCA cycle, exists on the inner membrane of the mitochondria. So, if the succinate dehydrogenase producing succinate continues through the TCA cycle, we can get oxaloacetate. Oxaloacetate can continue through the TCA cycle and become acetyl-CoA again, or it can become aspartate or malate. It can become malate via malate dehydrogenase, and it can become aspartate via an aminotransferase reaction. Either way, becoming malate and aspartate allows for oxaloacetate to leave the mitochondria, be converted back to oxaloacetate, and then via PEPCK, or phosphoenolpyruvate carboxykinase, become PEP and undergo gluconeogenesis. Gluconeogenesis, whether it be via PEP, glycerol, or galactose, or amino acids, or lactate, occurs in the cytosol. So let's reorient ourselves. We have our mitochondria. I also want to mention, now that we've talked about gluconeogenesis a little bit, pyruvate, when PDH is inhibited, can actually become oxaloacetate via pyruvate carboxylase. In this situation, often it will undergo this process I just described, being converted to malate or aspartate, oxaloacetate again, and gluconeogenesis. Often these are times when blood glucose is low. So I very briefly talked about this complex 2, and this seems like probably the most complex area of the drawing. And so here we have our complexes representing the electron transport chain. We have complex 1, 2, 3, and 4. And so these complexes, which exist in the inner membrane of the mitochondria, pump hydrogens from the matrix into the inner membrane space. They pump the hydrogens against their concentration gradient utilizing electron flow. And then the hydrogens flowing with their concentration gradient can go through complex 5, or ATP synthase, producing ATP. Now, it's important that too many hydrogens don't get pumped into the inner membrane space. If too many hydrogens get pumped into the inner membrane space, something called static head occurs, and there is no more electron flow, and respiration comes to a stop. And so, to prevent this, there are many different mechanisms, one of which is the ANT on the inner membrane of the mitochondria, in which ATP gets pumped out into the inner membrane space, ADP gets pumped into the matrix, substrate for the ATP synthase, and hydrogen ions get pumped back into the matrix, helping to maintain the membrane potential. In addition, another mechanism for maintaining the proton gradient is this hydrogen ion and phosphate symporter. This phosphate also is helpful as a substrate for the ATP synthase. And lastly, we'll talk about NNT real quick. NNT actually pumps hydrogen ions from the inner membrane space. I drew this a little poorly, makes it look like it's coming from the cytosol. But NNT pumps hydrogens from the inner membrane space. Using this energy, it actually can convert NADP to NADPH. This NADPH is actually really helpful for metabolism of reactive oxygen species. It helps by reducing the TRX, PRX, glutathione reactive oxidative species metabolizing pathways. And so, we have the electron transport chain, but where I mentioned that what's driving this hydrogen ion flow is electron flow. Where are these electrons coming from? And so the electrons can be coming from the TCA cycle. Remember, NADH and FADH are electron carriers, and they are both produced in the TCA cycle. It can also be coming from the cytosol. And so in glycolysis, you can get yourself some NADH. And so how do you get the electrons from the NADH into the mitochondria? You can do that via the malate aspartate shuttle. So the malate aspartate shuttle basically reduces malate. Malate can enter into the mitochondria and then become oxidized, and that allows electrons to be transferred into the mitochondria in very simple terms. In addition to that, you have the glycerol 3-phosphate shuttle, allowing for the electrons on FADH to be transported into the mitochondria. So, the one thing we haven't talked about yet, see if I can focus better, there we go, is fatty acid oxidation. And so that's up here in this corner. You have fatty acids via acetyl-CoA synthase being converted to acyl-CoA in the cytosol. Acyl-CoA via CPT in the intermembrane space can be converted to acyl-carnitine and then reconverted to acyl-CoA in order to enter the matrix of mitochondria. In the matrix is where beta oxidation occurs, and that creates acetyl-CoAs that can enter the TCA cycle. And so, in summary, we have a lot going on here, but in the cytosol, we have creatine kinase and adenylate kinase. But both of these enzymes exist in other compartments. Creatine kinase can be found in the inner membrane space. Adenylate kinase can be found in inner membrane space and the matrix. As far as glucose metabolism goes, glycogenolysis and gluconeogenesis occur in the cytosol. Glycolysis occurs in the cytosol. Pyruvate conversion to lactate occurs in the cytosol, but also may occur in the inner membrane space, LDH, as well as the matrix. pyruvate and lactate can enter the matrix of the mitochondria or the intermembrane space via MCTs. Pyruvate can also enter via these pyruvate hydrogen ion symporters, these pyruvate hydroxide antiporters. Furthermore, the TCA cycle occurs in the matrix of the mitochondria. However, citrate can exit the mitochondria, where fatty acid synthesis and triglyceride synthesis occur in the cytosol. The electron transport chain occurs on the inner membrane of the mitochondria. Oxaloacetate can leave the mitochondria via malate or aspartate to undergo gluconeogenesis in the cytosol. We have many different methods of transporting hydrogen ions in the electron transport chain. I talked about these different pumps and how they pump hydrogens into the intermembrane space. But hydrogens can also be pumped out by this ATP synthase, this ANT, and this hydrogen phosphate symporter. All three of those are responsible for pumping hydrogen ions back into the matrix. And lastly, we have NNT, which also pumps hydrogen ions into the matrix and also helps reduce NADP to NADPH. And NADPH has important reactive oxidative species roles. So, I hope you learned a little something from this video. If not, I'm sure you've already seen videos by Nick Vierhoven. His videos are pretty impressive, and I'm sure they're a lot more well-drawn than mine. He actually has a YouTube channel and everything, but at least I got my extra credit for my class. So, thanks for listening, and enjoy class. Bye.